Hello, thank you all to our monthly seminar from the Iberian Group of Urban Ecology. Today we are very excited to have here Dr. Paula Matos, who is a researcher at the University of Lisboa, specifically at the Institute of Geography and Spatial Planning. I think this is the translation in English. I also speak in Portuguese. So Paula did her studies first in Coimbra, biology and ecology, and then she did her PhD um, in the Universidade de Aveiro on global chain biologia e ecologia das alterações globais. What I've seen. And she works a lot with lichens, with vegetation. I just recently discovered that she's really involved in Arctic research. So she visits very often the research stations in Antarctica, which is pretty cool. As well as she does a lot of urban ecology. We had some common projects in Europe with different lichens, and also a lot of functional approaches or multidimensional biodiversity, also ecosystem services and functions. So we're very excited to learn more about your work. Thank you very much for joining us, Paula, and a pleasure to meet you, not in person, but at least virtually.
Almost in person.
Exactly. The floor is yours.
Okay, so I'll share the presentation just to make sure that you can. Okay. Can you see the presentation in full screen?
Yeah.
Okay. So thank you so much for the invitation to be here. It's a pleasure. So what I prepared is kind of a roadmap to showcase a little bit of what me and the rest of the group, because this involves a lot of people, including Thulema, that I think I saw her here. So I'm also showing a little bit of her work just to show you how lichens can be a window into urban ecology. So we're going a little bit from air pollution and climate to ecosystem services and health risk assessment. So to start, just a little bit about me. I've told a little bit, but I'm a biologist. I have a master in ecology and a PhD in biology and ecology of global change. Currently, I'm a researcher at the Institute of Geography and Spatial Planning in the University of Lisbon. I'm also the co-coordinator of the group, the research group that I'm in here in the Center for Geographical Studies. I'm also involved in the direction of the Polar TUI, which is a kind of a school that brings together different schools from Lisbon University, all working in extreme environments, because of that part of my work that is related to Antarctica. And I'm also a member of the ethics committee here in the faculty. So about the work that I do. So basically what I do is I study the relationship between organisms and their environment, and with two main focus, I would say, right now. So one is a biogeography perspective. So I study how species or how biodiversity as a whole, in its main forms, taxonomic, functional diversity, etc., distribute according to climate. And the other is to use biodiversity as an indicator of global change effects. And that's where my work in urban ecology fits. Since 5 years to now, I've been working on Antarctic, and I study the effects of climate change on terrestrial ecosystems. And the link is that most, as you will see, a big part of my work is working with lichens as a tool. And the big part of the vegetation in Antarctica are lichens, so that's how the bridge was done. So why have we been studying urban ecosystems? Well, but this is for an urban ecology group is a little bit weird, but it's just to contextualize our focus or our work with lichens there. So the main idea is that cities are very dynamic, and they suffer a lot of pressures. And in that sense, it's important to assess how ecosystem is functioning across several spatial gradients. And to do that, we need a kind of a link in urban ecology. So urban ecology, and in my perspective, using lichens, help us understand a little bit how this build structure and all the other global change drivers that we have acting on cities influence what we have, the ecosystems that we have on cities. And this is both from the perspective of understanding how ecosystems are doing, and with the other perspective also linking to us, people living in cities. So basically, it's to understand how all these factors shape biological responses. Urban cities are very complex systems. They have strong spatial heterogeneity in the sense that we have very different built structures, different types of vegetation, different land cover. We have multiple co-occurring drivers, including, of course, the most, I would say, pressing ones in cities, which are air pollution and the urban heat island, which is here represented by temperature, moisture, etc., and other kinds of disturbances that act simultaneously. And then in cities also at the same time, we have processes operating at very different scales. So we can look at the city as a whole, we can look at the city as part of a regional gradient, but we can also look at the cities from a very micro scale. It's like in this image, imagining that we were looking to what is happening in this avenue. And then another thing that it's important is that ecological responses are often non-linear and very context depending. And that is also a challenge in this kind of urban contexts when we are working with in the urban ecology. So for that, we need tools that are able to integrate all these complexities. So we need ecological indicators that are able to both respond to this complexity and to help us understand it. And so lichens come as a tool in this sense. So why lichens? Lichens are very powerful ecological indicators. They have a very long history as ecological indicators, and this is basically because they respond to atmospheric conditions very directly. They integrate the environmental signals over space and over time. They reflect both pollution and climate, which is very important in this urban context in these days. And they occur across a wide range of urban habitats. So that means that they have all these characteristics that make them good ecological indicators. To understand and to tell us a little bit about urban environmental change. And what are lichens? Just for those not so familiarized with lichens. So lichens are a symbiosis between a fungus and a photosynthetic partner. And unlike, for instance, plants, vascular plants, they have no roots and no cuticles. So that means that they absorb everything directly from the atmosphere. As you can see here in this video, this is a lichen hydrating, what occurs when you hydrate the lichen. So basically they are like sponges. They are strongly dependent on atmospheric inputs for water and nutrient supplies. And that means that they are very suitable ecological indicators of environmental change related to atmospheric conditions. So how do lichens come up in urban ecology? So what is the scope of this work? So basically, in first place, they are what I'm going to talk about. today is how they can be used as ecological indicators of several environmental drivers that occur in urban contexts. Secondly, how we can use ecological indicators across different urban scales. So connecting a little bit to what I've said about the urban complexity. So from the micro scale of an avenue to broad scale, imagine European urban contexts. And then how we can use these indicators to inform us about ecosystem services, risks, and use them as a planning and management tool at the city level. So starting with Lichens as urban environmental change indicators. The first thing that we should pay attention to is, first of all, think about the drivers that we have in cities. So these graphs here depict some that used to be historically, so SO2, sulfur, it used to be a really big problem environmentally, but it has been decreasing since here in Europe, since the legislation, the European legislation for regulation of these pollutants started, and it has been decreasing sharply. But for instance, we have other drivers like ammonia that still are increasing. This is not a huge problem in cities, but other nitrogen-related pollutants might be a problem in cities. And of course, we have then climate. We know that climate is changing and will continue to change, and we need also to be able to, under an ecological indicator perspective, use Lichens for that. So this means that we have different environmental drivers. Each of these drivers have different intensities and different future trajectories. So we should pay attention to this when we are trying to use Lichens as tools to indicate these conditions. Because different urban pressures, of course, will lead to different biological signals. So the first thing is to think about which kind. We can use Lichens in several different ways. And of course, when thinking about working in urban context, we need to think first how to use them. And that immediately passes from thinking what would be the best metrics, biodiversity metrics. We can go from physiological stress to species loss. And these indicators, of course, change with intensity. So just to give an overview, if we have a low intensity environmental driver, physiological performance will probably be enough. We will see the sensitive species having a decline in their physiological performance, and then tolerant a little bit further on. So we can use the threshold for individual performance and ecophysiology-based metrics. But then if actually the driver is a little bit more intense, what we will see is that we will move to the species level. So some species will start declining, the more sensitive, the tolerant will still hold on a little bit, and then will decline as the intensity of the driver increases. And we can use also these thresholds and trait-based metrics, so functional approaches to depict the effects of these drivers in cities. But then if the driver is really very intense, and a classic example of this would be high intensity pollution, we will see that species abundance declines. And then if it's really too intense, we will see a decline in species richness. And in this sense, taxonomy-based metrics will be the best. So this is just to make an overview of the different types of metrics that you will see along the story that I'm going to tell today. Each of the metrics is selected according to the type of driver or the type of the intensity of a driver, because not all of the metrics work with all of the drivers. what it is important is to understand that the stronger the pressure, the simpler it is, the sign actually when we are using lichens. And I would say that probably with all the organisms, I think a decline in species richness is one of the easiest messages to convey when we are talking about the impact of some kind of environmental driver. And this leads me to the first work. So here we are trying to detect. We used lichens to detect pollution, but that specific pollution. So this work was done in a small city here in the south of Portugal, so in Sines, and we were interested in with using lichen diversity to see if we could detect at the city level. And you can see this in the map. So we've mapped all the houses and traffic, the roads, and the green spaces to see if lichen diversity at the city, in a small city like this, so with low traffic pollution, would be able to detect the low traffic pollution sign. And in fact, we saw that traffic pollution changed, in fact, lichen communities. And in this case, we use a functional trait approach. So we separated lichens according to several traits. I'm just showing here two of them. So according to the tolerance to eutrophication, which is this first graph, and then because pollution tends also to, so I forgot to say, so this is all of the work that I'm going to show you is done with epiphytic lichens. So we see the lichens that are growing on trees, more or less at breast level, because these are the best to capture the signal of the atmospheric pollution. And because pollution sometimes tends to change the pH of the bark of the trees, the lichens can be specific. Some species of lichens like more acidic conditions, other like more the opposite conditions or more neutral pH. And we could see here, you can see here in both graphs that in traffic, so near roads, we always have the lichens that are more tolerant to pollution or more used to basophilus pH, so basic pH on the tree, both associated with traffic pollution. So sensitive groups decline near roads. We have an increase of the tolerant groups. And in this case, lichens were really good as indicating traffic related pollution. But the important thing is that we are talking about low levels of pollution. So this is a small town compared, for instance, to Lisbon or other big cities. In another case, we tried to understand if this would also work in a subtropical cities. So the idea here was to using a similar method also with epiphytic lichens, so lichens growing on trees, on tree trunks, to see if lichens would be able to distinguish these different cities that you can see here in the map. This is in Rio Grande do Sul, so it's a subtropical region in the south of Brazil. These cities are different, so they differ in the kind of industrial pollution that they have and the kind of urban pressures that they have. And you can see that, so each of these circles represents a different sampling site inside of each city, represented here by the squares. And the colors around the central dots, they represent the different land covers, which are proxies in this case for the different type of urban pressures that we can see in each city. So what we saw is that in this case, two things were done. So first, we went for, and this now remembering what I said before about the metrics. So in this case, we used also ecophysiological metrics. So we see the physiological performance in the lichen, basically measuring photobiont vitality, photosynthesis, how the photosynthesis is doing. And we could see that the more the polluted the sites were, the lower the photobiont vitality. And then the next step was to use the species diversity, the community composition. And in this case, the community composition is here represented by the colors inside each of these dots, and they come from an ordination technique. And we could also see that also the community composition was responding to the different levels of pollution in the cities. So in this case, even in under different conditions in a subtropical region, lichens were able to reflect both the pollution and the urban pressure together, showing us the complete gradient of urban pressure in these cities. Then we have a classic problem in cities, which is the urban island. So I think I'm guessing that this is well known for all of you. But urban island is an effect where cities have, on average, two more degrees than the surroundings of the cities. And this is a work also in Brazil. There are also others from our team here in Europe, but I thought that this, as this was the first time that was done in a tropical place, it would be more interesting to show. So we also wanted to know if lichens were able to signal this urban island effect in the city. So again, same methodology. We go on trees, we sample lichen diversity, epiphytic trees, and here the sites were categorized into two classes. So whether it was low urbanization with the highest percentage of forest cover, or medium-high urbanization, which means more dense and more dense urban tissue, so with more houses, more roads, and less green spaces. And then different types of functional traits were used to apply functional structure metrics in this case. And what we found was that urban sites, they differed in the built-up intensity in these two categories, and that in more urbanized areas, lichens indicated that the environment was warmer and drier, the typical urban island effect, and this was shifted towards low urbanization sites where we could see a predominance of lichens with traits more water related. So basically species that need more water to survive. In this case, it was clear that urbanization created a microclimatic filter that filtered the species that live there, thus allowing us to use lichens to indicate the places where the urban island could be observed. Of course, this was seen because here pollution is at a very low level, and the urban island was easily seen. But you will see further on in the presentation that when pollution levels are too high, seeing the effects of climate or microclimate could be much difficult. Then this is another work here in Lisbon, where we tried to see if we could use also a trait approach and measuring lichen diversity in 42 green spaces in the city to see if we could distinguish the different places according to the anthropic disturbance. So in this case, we used the idea was to test a specific trait called poliotolerance, which is not frequently used with lichens, though it exists in the database, and to see if we could distinguish if lichens using this trait could give us an idea of a disturbance as a whole, a disturbance gradient in the city. So what you can see here in the map is the blue areas. They are sites with lower disturbance, and actually this big blue here, the blue part here is a forest that we have inside Lisbon, Monsanto. And then you can see the high disturbance places here in the city center. And I would like you to pay attention to this particular place here, because this will be the one of the micro scale level works that we did inside Lisbon further on. So what this work shows us is that species tolerant to disturbance increased in places which have higher anthropic pressure. So basically this lower air quality or higher disturbance is due both because here the urban density in the center is bigger, but also because we have a lot of traffic. So this area here is seen as the most polluted area in the city. So in this case, lichens were good to indicate this general disturbance gradients. Then we tried to see high intensity pollution within the city. So in this case, it's again Lisbon, but we were interested in trying to see how good they were to show us air pollution, in this case PM10, so particulate matter, in the city. To do that, of course, in the places where we have the air quality stations, lichen diversity was calibrated against the concentration of the PM10. And then using models, we modeled the distribution of lichen diversity to give us an idea of the air quality in all of these green spaces that you can see here with these 5 colors around town. Again, the blue, this is the forest that I was talking before, and this is again the lowest air quality region in the city center that was showing up here and here in the other image. So we saw that Lisbon has, in fact, still, though the pollution is lower, we still have a strong pollution gradient in the city, and that shows what it translates to it, that lichen communities get simplified. So we have a species richness sharp decline, and this is still the best indicator to use in this case. So lower lichen species richness, lower air quality in town. Then the next work is to show you a little bit how we use lichen indicators across urban scales. So the first case, this is from Zulema. I think I saw her before. I don't know if she's still here. This is her work. that she did while she was when she came working here in Lisbon a few years ago. And in this case, this is a nice example to show how moderate pollution can be highlighted across broad scale gradients. In this case, it's moderate pollution, so several cities were compared. Pollution gradients were more moderate than the pollution gradient that you saw inside Lisbon. And urban conditions were very different among cities. And in this case, unlike the species richness in Lisbon, because we have a little bit lower pollution levels, so the total lichen diversity was the best indicator. It could actually depict the differences between across cities. The following work is one of those works that me and Joan were together in an European project. So this is a broad scale climate and pollution gradient. And the idea here initially was to try to see if we could see the climate signal, because it is important that our lichens, given that we have a climate change problem these days, that lichens are also good to indicate climate. But as you can imagine, when you have several interacting drivers, in particularly when you have still high pollution in cities, that may in turn difficult the getting the climate signal on top or in spite of the pollution. So the idea here was to sample again lichen diversity in several cities across Europe. So from Almada, which is just across the river here in Lisbon, to Tartu. This encompasses a very wide climate gradient, but these cities have also different pollution background. So this is also a pollution gradient on top of the climate gradient. What we did first was to use the conventional approach. So basically all of the works that you saw before, on their basis, they always have some kind of a linear model approach. And what we saw was that you can see it here in these graphs, was that even using different biodiversity metrics, so from taxonomic, functional diversity, or functional structure, using an array of different traits related either to pollution or to climate, you can see that the first thing that comes to our that comes up is that the models were explaining a very low portion of the variance, and that's what we wanted, which is climate, which is again together the red and the blue, so temperature and precipitation, are a very, very, very low portion of that when we partitioned the variance of the models, the linear models. And pollution is still within the variance explained, is still the main driver, regardless of the metrics that we used. So what we can see in this case is that air pollution still gives the clearest signal. And this is a problem because if we also want to use climates to signal climate change, lichens to signal climate change, this approach was clearly not working. Then the rest of the variance here not explained was related to local conditions. So what this means is that the traditional statistical lens, or the conventional approaches that we were using before, were clearly insufficient to enable us to see the climate signal using lichens. So next we tried a different approach. We went for non-linear a nonlinear approach. So what I'm showing here is the result of, in this case, we didn't use linear regression. So we used a nonlinear approach, random forest regressions. And then what we are seeing here is that instead of running just one model, we ran an ensemble of models. So this is the median of 100 random forest models for each metric that we use. Again, the same metric. So we use several taxonomics, several functional diversity, and several functional structure metrics using the same traits as before. So traits that we know that respond to either climate or pollution. And we can see that here the picture changed a lot. So with this new modeling lens, we could see that the models performed better. And if you remember from previous ones, so climate was just a very little portion of the variance explained, while here climate, which is the sum of blue and red, were the main ones, regardless of the metric that we were using. And that is very interesting. So not only climate was the main, as when we go see the individual responses for these models, we could see in most of them. So this is different metrics. You can see here on the left. And on the top, you have the climate variables or pollution variables that were used. You can see that for many climate variables, like for instance, this bio12 total precipitation, you can see a threshold associated with the lichen response, which is basically what we are looking for when we use lichens as ecological, when we are using an ecological indicator. So these nonlinear models revealed important climate thresholds, which is a very good thing. So in this sense, what we saw is that lichens can be, in fact, good to disentangle the effects of climate and pollution in an urban context. And then we go, this was at a broad scale. And then last year, we thought about, we had, so in this, the place that I'm working now, a few of my colleagues, they were doing these models, which are shown here where I'm having the mouse now. So they do, this is Avenida da Liberdade, so this is an avenue, that one that I showed before that was always one of the most polluted sites. So this is seen as the most polluted street here in Lisbon. And they wanted to model what's happening. Why is this road so polluted? So this is a road that forms like a tunnel of trees, like you can see here on this image. It's kind of an urban canyon. So this is a street that goes down. Basically, if you go all the way down, you would find the river. So that means that you will have the wind flowing in. It makes like a tunnel, a wind tunnel effect. And what they did is using some models, fluid dynamic models, to model pollutant dispersion and wind flow in this canyon. And when they did it, okay, they got to these models, but then it's very difficult to actually corroborate the models, because you have a air quality station, but you have only one, and it's here and at the bottom of the street. And so one day I thought, okay, let's try to see what I can say. So what we did is on 82 of these trees around, so there's like 4 rows of trees here. So all of these green dots here, we sampled lichens. And and lichen diversity again on the always using just one type of one species, so Celtis australis, and we measured the lichens growing on tree trunk. And then we did a multivariate analysis and mapped it to see what actually lichens were showing. So the community shifts were showing. And actually what we saw is that if you pay close attention to what is seen. So this is axis, the first axis of the ordination, and then the third axis of the ordination. You can see that it's actually mimicking both the wind patterns and the pollution patterns. So these are two pollutions, NO2 and PM10. We can clearly, lichens clearly show that the left side of the avenue, it's much more polluted than the right side. And it's also different in terms of microclimates, which shows that they can go to this micro scale level. So we're talking of just one avenue. And so they actually corroborated that the canyon conditions are highly heterogeneous and help validate their physical models. And this brings me to the last part. So it's how do we move from the indicators to ecosystem services, risk, and planning. And the first work that I'm showing here, it's related to human health. So this is a work that was done. So here, basically, we sampled lichen species, a specific species that was growing in this place. So this is here where I'm passing the mouse. This is a Foodness. This is a very small town or village in Azores, so an island from Portugal in the middle of the Atlantic. This is a very volcanic region. And what we wanted to do is to try to see if lichens were able to show us the risk of exposure to hydrothermal volcanic emissions. And so what we did is we collected we placed lichen transplants. So basically, lichen transplant is you collect specific species that are good for this from clean sites, and then you expose them in these places for a certain amount of time, and then you analyze for the contaminant that you want to see. In this case, we analyzed for delta 34S, so a stable isotope for sulfur, which is a signal of the emissions. And so how do we pass from the concentration that you can see here of the stable isotope that measured on lichens and the distance to the closest fumarolic field. So this is a very, as I told you, active in terms of fumarolic activity. And you can see that the concentration of the isotope differs with distance. But being that the more negative the concentration of the isotope, the closest you are to the emission source. So after we get these results, so we know that they are good at depicting the signal of the fumaroles, we can use these to map the risk. So how do we do that? So first, we use the lichen concentration as the hazard. We map the hazard, and then we see the exposition. The exposition is done, the exposure is done by seeing, okay, where do we have the houses? That means that if we have a house, the emissions, the hydrothermal volcanic emissions from fumarolic activity tends to accumulate inside houses, and that's an extra danger for people that live inside the houses. So afterwards we see what is the exposure, so we map the houses, and then we cross that to the population distribution, because of course we can have an hazard, but if there's no people living there, that will not translate to risk. So the risk is in fact a mixture of these three different things. And then what this is used to support territorial planning, so to inform. Here in the map you can see, so the dark areas are the areas of very high risk, where the risk is higher for exposure to this, because this has several impacts on people's health. And in this case, you can see how this to show you a little bit how lichens can also be used to make spatial explicit models identifying chronic risk areas, in this case for health. Then we can also translate this into more practical management guidelines. So in this case, it's also in Lisbon, where we saw that lichen, the high intensity lichen. Then we cross that information with the green spaces sized. So here we are talking about the services provided by urban green spaces in terms of air quality improvement. And then we cross that with the size of the green spaces and the density of the green spaces. And what you can see here, without going into much detail, is that we can translate these models into showing the municipality how much they can gain into what would be the increase in air quality if they increase the area of the green space or the density of the green space, or a mix where we increase 50% of the area and 10% of the vegetation. What would be the gain in terms of this percentage shown here? And this for three kinds of green spaces. So from small to large green spaces and low density to high density green spaces. So in fact, this shows how the information collected with lichens as ecological indicators can be used to identify priority areas for management at the city level, in this case relating to air quality regulation. But we also saw that we can do that. Then we went also to see, okay, but are all green spaces the same? I mean, do they all provide the same services? And we saw that no, not all green spaces are equal. So this was done in a big green space, not here in Lisbon, in Almada, in another city. But it is the very big green spaces that have different vegetation structures. And here the idea was to assess how air quality and climate regulation, the ecosystem services of air quality and climate regulation varied with the vegetation structure. In this case, woodland, a natural woodland, performed better than lawns. So woodlands are here indicated by the dark green. And we also cross-checked, we model also the management that is done in each of the areas. And we saw that the management also influenced the ecological function that these green areas had. So overall, green space quality mattered. And in this case, not only area, meaning that it's not, for instance, the same to have, let's plant green spaces to have better air quality or to regulate the climate, to lower the temperature in cities. But planting a lawn wouldn't do, for instance, the amount of service provided by a lawn is really, really low. So we need to think that if we want to do this, we should think about what kind of vegetation structure we should have and its composition, because they are keys to optimize these ecosystem services at in urban at the urban scale. And then as a synthesis of all this, So what I've likened in all of these works that I've showed you from everyone that I worked with over these years revealed. So urban change is multi-driver and it's complex. And the environmental signals that we see sometimes depend on the scale. The responses clearly depend on the pressure intensity. Indicators can connect both the ecology, the risk, and the planning. So we can use these both from a biological perspective, but we can also apply it as a tool to help manage cities. And the important is that key message is that urban change is not one signal. It is a layered environmental story, and it should be seen as such. And then what are they good for in the end? For air quality gradients, for the urban heat island, so heat and dryness, for anthropogenic disturbance, for pollution thresholds, microscale urban processes, and ecosystem service provision. And of course, then in the end, we can try to use some of these to calculate exposure, vulnerability, and risk. And basically, lichens make the invisible urban pressure effects biologically visible. And that's it. Thank you. Stop sharing. I'm sorry if it was too dense. I tried to synthesize everything.
No, it's super good. Thank you for this super good presentation. There is a lot of science behind, a lot of research, a lot of nice results. I'm surprised about the high resolution of some of your results, like really in the tree almost, no? Like
we tried, that was my last one, and I said, okay, let's just try. So I just went to check one or two trees because I didn't want to spend a lot of time doing and then not seeing, but I could see that they were different.
Yeah.
In one side and the other. So I thought, okay, let's do it. And I was also amazed. But we always have a joke, I don't know if you had it in Spain, but we had like this commercial in TV that is like, oh, the cotton doesn't tricks you. That they were cleaning and then they would pass the cotton, white cotton, just to see how clean it was. So we always have a joke in Portuguese, and in English it doesn't sound that good. Like, os líquenes não enganam. See, algo como los líquenes no te engañan. And they can always tell you, I'm always surprised because even when you think that they won't give you an answer, and it's kind of surprising how good they were to depict the physical model that they had. So they knew that something was happening because you have I couldn't explain all the details here. You have like a kind of the wind makes a tunnel and then it flows, then the trees do not allow the pollution to go out, so they stay inside. And actually lichens showed it perfectly. And I was also impressed with the resolution that you can use them to see it.
Super nice. So we have time for questions. I will let anyone who want to answer or to get some questions done. And if not, I have two small questions that maybe meanwhile I can make, if you don't mind.
Yes, sure. Go ahead.
So one is super small, but maybe you explain it and I didn't get it. What's the difference between what you call functional diversity and functional structure?
Okay, so it changes with schools, the functional diversity. So functional diversity used to be like the big. So if we think about the DIAs and the first papers, would be the name, the umbrella name for everything. But the most recent works, they separated into two. So basically, functional structure is when you're using community-weighted mean of traits.
Okay.
Or categorical traits, whatever trait it is.
Yeah.
And functional diversity, they only apply to if it's like raw, the functional richness. So the compound indices. Functional structure is just community weighted mean, and the rest is functional diversity. But so this is again it depends on the school that you are following.
Yeah. Okay. So ecologically is more meaningful the community weighted mean because it tells you more about how the community is, and the other is more like less. So you can extract less information, no, about the
I would
responses of
say that the difference, it depends on the question.
Yeah,
So
right.
imagine in this case that we are talking about cities, I always think that community weighted mean is easier to explain, because if you
Yeah.
want to translate this as like when we were saying, like, see here it's really polluted, so you need to shift the way the traffic goes, for instance.
Mhm.
And then you have to explain why you are saying that. So it's easier to explain, oh, you see the proportion of sensitive species decreases with pollution. So it's easier to say something like that, and it's very difficult to explain a RAW or
Yeah. No, that's what I mean. That
but
it's
RAW
more
is
easy, yeah.
good more ecologically. So imagine if we are talking about we were calculating how diverse the communities are functionally, then RAW would be the best one to see, because if you use all of the traits that you have available that make sense for your work, the RAW multi-trait RAW, not because you can also calculate single trait RAW, would be the most the better one. I would say that it depends, and it's just a way of how you name it.
Okay, and related to this, how you communicate to those results. So I guess the community weighted mean, so the functional structure or results are more easy to explain to the people. How is your experience with this trying to explain those results outside of academia?
We always think about that when we are testing. So I didn't show, but of course, for almost all of those works, we tested a huge array of metrics.
Yeah.
So if the idea is like in this work from Lisbon that we had, like, how do we improve their quality, improving size or whatever, we always think we have to think about the simplest way to show it. So if species richness is responding, it's always the winning one, because it's easy to convey the message. But for most of low intensity drivers, species richness does not work. So we do it like that. If community weighted mean is as good as RAW-Q, for instance, as functional diversity, I would go for community weighted mean.
Yeah.
Because it's easier to explain. You see, you have the sensitive species, either if it's for climate or the species that like more humidity conditions, you see, so you can see the urbanite island like this. But it is easier. It is easy to, but you need, we never go with all the array of metrics that you calculate. So you need to go with just one.
Yeah.
That's my thing. It is we should always, if you want people to listen to you and to use your data to actually do something, you should always do it like that. And that one that we saw that the vegetation structure was different, was showing different air quality and climate regulation ecosystem services. That one was passed to the municipality, but we did it in a very easy way. So it was using that community weighted mean.
Cool. Thank you. Maybe there are more questions in the chat or the people. I don't get Sulema if she has any question or not. I'm not sure.
Sulema, you can talk.
Yeah.
I forgot to say, but of course, this is the work of many people, as you could see in the authorship, because this is all published works. Zulema is one of my coworkers. Joanne is also in 2 of the works. And so, of course, this is not me. I'm just representing the whole team and trying to tell a story of what the whole work we do. Okay. You can't.
Yeah.
So maybe you can write
I think
it.
Zulema was asking a bit about your current work in Antarctica.
What I'm doing. more in
But
detail.
you just explained
Okay.
us before.
Sorry.
Okay.
I didn't get this. Sorry.
So what I do is I study the vegetation, the whole vegetation. So it started in the beginning 6 years ago when I got my first polar project. It was using lichens as ecological indicators of climate change in Antarctica, and it has been evolving since then, because in Antarctica the vegetation is mostly lichens and bryophytes. So I also do bryophytes in Antarctica and the 2 vascular plant species. We only have 2 vascular plant species in Antarctica, so it's quite easy to identify. They are easy. And so basically what I do is I study, I study the vegetation, so all of them, the 3 groups, along spatial gradients of climate, or microclimate, because microclimate is a very different thing in Antarctica. It overshadows macroclimate, because the conditions are so extreme. And I use it to see how ecosystem is functioning. I have a project now with the species distribution model, so the dominant species, because we still haven't it hasn't been done for the vegetation yet. So I do high resolution species vegetation models for the Antarctic Peninsula. I study the feedbacks between vegetation and permafrost, because now I'm working with geographers, and the permafrost team is here with me. So we also do this thing of trying to see both works, and I think that's the big thing. So not so much focusing on ecological indicators in Antarctica, but seeing from the other perspective, but also with an idea. So all of my work fits the Antarctic Treaty to help to try to give guidelines for better conservation efforts every year.
The recommendation would be to not put cities in Antarctica, no?
It is forbidden. So even if we wanted, Antarctica has no human, but yeah, the pressure from tourism now is increasing. So
Yeah.
we also are aware of that, and I also try to see it in my work when I do it, but I always try to go to places where tourists don't go. So, because I want to see the effects of climate, so not the rest.
Okay, so if there are no other questions, I think we can close the seminar. Thanks a lot, Paula, for this amazing talk and all the work you made and all the work you're making. And we will follow your updates in the future. We are super happy to have you here today. We need more people from Portugal in our seminars, because we are a little biased towards Spain.
You're bigger, so.
No, but I think it's because of we belong to the Spanish Association of Terrestrial Ecology, so I guess it's normal.
But it's linked with SPECO, so the Portuguese one.
Yeah.
So maybe you can try some common efforts to have more Iberian, more Portuguese representation. And thank you all for the invitation, and for all of you that were present.
Thank you.
Nice to see you,
Paula.
Zulema.
Bye.
Bye. Thank you.
I will stop the recording.
Bye, Nuno.